Hello everyone, I am Dr. Sathya and I'm here to give you an overview on the legal and regulatory compliances in new ventures. Before we start, let me give you a small introduction about myself. I'm a consultant toxicologist and I'm an American board certified and European registered toxicologist with more than 18 years of experience in preclinical testing of chemicals, including drugs, pharmaceuticals, medical devices, etc. I've been into the field of regulatory toxicology for more than 15 years now. I've had the opportunity to interact with various regulatory bodies like the FDA and the CDSCO of India. So I would like to share some of my inputs and the regulatory and legal requirements of a new product or a thought that would come across your mind in the near future. In the field of science, innovation and discovery is a never-ending process. Rather, it is a continuous process. Now, as medical professionals, you may explore and end up with a new drug or a new on state-of-the-art medical device or even a new cosmetic formulation, which will be helpful for, say, complex diseases or for those for which there are no cure at the moment. Now, the challenge is the thought has to become an action. The action that is your product has to come to the market. How does this happen? Well, this is a cascade of events that happens, and it is stringently regulated by regulatory bodies which are which differ from country to country, and they follow very stringent regulations so that the product that comes to the market is of high quality and is safe to its end users. Talking about regulatory bodies, the Central Drug Standard Control Organization is India's national regulatory body, which stringently controls the registration and marketing of cosmetics, pharmaceuticals, and medical devices. In the United States, this is handled by the Center for Drug Evaluation and Research. The very purpose of having such regulatory bodies is to have safe and effective drugs, medical devices, and cosmetic products. In terms of safety and efficacy, there is a series of tests which the manufacturer is expected to perform and document and submit these data to the regulators for a thorough review, following which the approval of the drug, medical device, or cosmetic is done and marketing proceeds further. This is a snapshot of the CDSCO of the Government of India, which has information on the various acts and rules which are dynamically updated and other information on the regulatory requirements for the registration of each type of products, etc. This page has information on the FDA CDER's rules and regulations, the laws that are enforced by the FDA, the most important documents that are the standards that the manufacturer is expected to understand, and other information on the freedom of information and dockets management. The products that are regulated by the FDA include food, drugs, medical devices, radiation emitting products, vaccines, blood, biologics, animal and veterinary, cosmetics, and tobacco products. Now, each of these has its own regulatory requirements, and each of these subsections leads to the respective regulatory requirements and the documents that are supposed to be annexed along with the main application. For the sake of demonstration, I wish to use this flow chart of the FDA on the drug development process. The foremost step that happens in the drug development is the discovery. If you look at the drug discovery process, initially about say more than 1 lakh molecules are identified as leads, but then based on the structure activity relationship and things, say about 250 compounds come to the preclinical development phase, where in vitro and in vivo studies are performed to understand the safety and efficacy of these molecules. Now, in vitro refers to tests that are conducted in lab dishes or petri dishes or other similar laboratory equipments. In vivo studies represents animal studies. Step two is the preclinical research, where we spoke about the in vitro and in vivo tests to answer basic questions about safety. When a drug molecule passes the safety studies, that has to be tested on humans, which is the step three, which is called as the clinical research. Now, when drugs have to be tested on people, it cannot be directly done. It needs the approval from the FDA, and there is a documented procedure for a clinical trial to be initiated. This document is called the Investigative New Drug Application. Now, in order to conduct trial in humans, there is an application called the IND, which has to be filed with the FDA before the clinical trial is initiated. Now, the IND is used to investigate the safety and efficacy of a drug in human beings. Now, an investigative new drug application is a request for authorization from the FDA to administer an investigational drug or a biologic product to the humans. Now, how does an investigative new drug application actually help the manufacturer? The current federal law requires that a drug be the subject of an approved marketing application before it can be moved outside its state. Now, because a sponsor may want to conduct investigational trials outside his own country, it has to be transported across state borders, for which an exemption has to be sought from the FDA as a legal requirement. The IND is the means through which the sponsor technically obtains this exemption from the FDA. Now, the FDA's role in the development of a new drug begins when the drug's manufacturer screens the new molecule for pharmacological activity and acute toxicity potential in animals, and then files the IND. Now, from a molecule, the the name technically becomes a investigational new drug after all the specific requirements of the drug regulatory system are fulfilled by the manufacturer. Now there are three major types of INDs which the manufacturer can choose from according to the situation. The first one being the investigator IND, the second one is an emergency use IND, the third one is a treatment IND. An investigator IND is submitted by a physician who initiates and also conducts the investigation, and he will be the sole responsible person under whose immediate direction the drug will be administered or dispensed. A physician may also submit a research IND to propose studying an unapproved drug or an approved drug for a new indication or a new population. The second one is an emergency use IND, which allows the FDA to authorize the use of an experimental drug in an emergency situation that does not allow time for submission of an IND according to the required regulatory requirements. Now, we have a typical case. During the COVID-19, drugs were approved on a fast track basis. They were called as emergency use approvals because in a pandemic, there was no cure for COVID at that point of time, and many people were dying on a day to day basis, on a minute by minute basis. So, in order to save the lives of thousands of people, the emergency use authorization was implemented by the FDA, and two drugs were initially uh, approved by the FDA, but unfortunately, remdesivir did not work as expected, so it was recalled. And Paxlovid is the one that is currently also being continued for the treatment of COVID. The third one is called the treatment IND, which is submitted for experimental drugs, which show promise in clinical testing for serious or life-threatening conditions. While the final clinical work is conducted and the FDA review is also taking place. And there are two major IND categories, they're commercial and research. Coming to the main content of the IND, what should the IND contain? The IND is expected to have three of these major things. First one being the animal pharmacology and toxicology study data, the manufacturing information, and clinical trial protocols and investigator information. Drug development itself is a long and expensive process that follows a series of sequential steps that we saw a few slides ago. What researchers initially do is screen for candidate pharmacological agents for desired activity and specificity, and then establish potential ranges of safe and therapeutic doses in the preparation for human clinical trials. Now, preclinical data permit an assessment as to whether the product is reasonably safe for initial testing in humans. This is what is collected during the animal pharmacology and toxicology studies. During the preclinical stage, as we saw earlier, the drugs are tested for their safety and efficacy. Now, recently, we are seeing that the biomedical research is undergoing a paradigm shift, which is centered on human disease models owing to the notoriously high failure rates of the current drug development process. So the major drivers for this transition are the limitations of the animal models, which despite remaining the gold standard in basic and preclinical research, they suffer from interspecies differences and poor prediction of human physiological and pathological conditions. Now, while we have the in vitro and non-human in vivo preclinical models being the integral component for being existing in the history of drug development, they frequently fail to accurately predict a drug's candidate's performance in the subsequent clinical trials in terms of efficacy, specificity, toxicity, or a combination of all of these. Currently, the drug development enterprise is facing an increasing challenge in the successful production of new therapeutics. These challenges are also in part due to the increasing costs which are associated with these drugs that passed preclinical studies but failed in subsequent clinical trials. As a result of these, there is a critical need to make advancements in the models available for preclinical studies. We see that approximately 75% of drugs that emerge from preclinical studies go on to fail in phase 2 or phase 3 clinical trials due to lack of efficacy or safety. So the reliance on non-human animal models in preclinical studies is a significant contributor to this failure. There are fundamental biological differences between small animals such as mice and humans, and this is a most frequent cause of failure to predict a potential drug's efficacy and toxicity. Large animal models such as pigs, dogs, and primates can improve the predictive value by introducing anatomies and physiologies that are more similar to humans. However, large animal models introduce a significant burden of cost, time, and increased ethical considerations. Even with improved predictive power of large animals, molecular, genetic, cellular, anatomical, and physiological differences do persist. As a result of this, we see that there is a significant demand for preclinical models based on human tissues. And talking about preclinical models based on human tissues, the bioengineered tissue models, the organoids, and the organ on chip are some of the significant contributions from the researchers. The bioengineered tissue models are generated from human stem cells or primary cells, the latter of which are isolated from surgically excised human tissues or from non-transplantable organs. These cells are then manually added or 3D bioprinted onto hydrogels or polymer-based scaffolds, often in transfill setups. They can also be used on decellularized ECM scaffolds from animal-derived organs or non-transplantable human organs. The advantages of these models includes the possibility to cultivate them at the air-liquid interface or submerge, thereby emulating an in vivo-like tissue condition and their maturated and differentiated tissue state, and achieving a increased biomimicry of native human tissue compared to the 2D models. Now, this approach is often used for multi-layer or stratified tissues such as the gut, lungs, and the skin, but also the brain and liver as it allows a controlled buildup of the model. However, these tissue models cannot be cryopreserved or propagated, and therefore they have a limited lifespan. And given their limited cell diversity and lack of self-renewal in non-stem cell based models, these did not click so well. The organoids, on the other hand, they are self-organizing 3D structures which are generated from tissue-specific cells. In these models, the stem cells are embedded in an extracellular matrix that acts as the scaffold for tissue growth. The organoid formation is then guided by a cocktail of growth factors that are important for tissue development in vivo and which also helps them to maintain the differentiation and self-renewal, which was the significant disadvantage with the bioengineered tissue models. Now, the size of these organ-like structures, ranges from 100 micron to 2 millimeter, and they can be cultivated in small dimensions. Thereby, they are also be suitable for high throughput screening. The most interesting innovation from the researchers is the organ-on-chip model. Now, when comparing to the other models, the intertissue crosstalk or complex in vivo like processes cannot be mimicked in single and static tissue models. To enable tissue perfusion and dynamics as well as multi-organ crosstalk, the organ-on chips were introduced. Now, these are perfused microfluidic platforms that contain bioengineered or miniaturized tissues or organs, which are interconnected by 3D microchannels in order to simulate the in vivo functions, the biomechanics, and also the physiological responses of the organs. These are often referred to as microphysiological systems because they can emulate human pathophysiology in a more human-like environment. Now, as opposed to organoids that form by self-organization, these organ-on-chip models follow a reductionist engineering approach through the targeted and predefined design of components such as the scaling of cell numbers used based on their physiological function or including disease-relevant cell types and key physiological and biochemical cues. Progress made in the field of human-like tissues for drug development and testing is quite an amazing thing. But it is very unrealistic to assume that the entire human with its given complexity can be fully replicated in vitro. At the same time, the ideal should be to reach a maximum level of clinical mimicry. Now, this is going to come with several challenges which will remain and which are still unresolved, and they are having their initial drawbacks. And there is a lack of cell diversity, vascularization, and the ability to study tissue crosstalk. All this have been taken care of, but still we have a long way to go when it comes to the regulatory acceptance of these new models. Now, regulatory agencies are mandated to protect human and animal health and the environment. Decision making about hazards and risks requires data, usually that includes toxicological test results. Now, research and regulatory agencies they develop and adopt test methods or strategies that ensure that toxicological data are scientifically sound, consistent, and usable in the risk assessment process. Now, this is where the in vitro models, the most recent one, can actually find their a very difficult process to go through. The consistency of obtaining results with these models could be challenging. Now, the regulatory acceptance of these new models is challenging because most federal agencies agree that, in addition to the specific requirements of the agencies, these new methods or technologies, they must meet certain minimum standards for validation. And the methods must be reviewed and commented by the interested public. Now, all this is going to take a lot of time. The, the purpose of developing on the one end and the time taken in implementing the same is too much that one day the technology may become useless for the end users. Again, federal agencies also differ widely in their procedures, and the international guidelines adopted by one federal regulatory program or agency is not necessarily used or accepted by the others. Now, this also creates the repetition of procedures and the repetition of validation process for each type of regulatory body. which makes the whole process all the more complex than it is actually expected to be. So we spoke about the animal pharmacology and toxicology studies, the new developments, and how regulatory acceptance of these methods could probably happen, and what are all the hiccups that are underway for these new methods. Now, the next component in the IND will be the manufacturing information. The manufacturing information that is required in the IND includes information pertaining to the composition. I mean, it's a very basic thing that is required in your application. Like, what is your drug made up of? That is, like, what is your main ingredient? What are the excipients and what are the impurities that are expected? And is everything, all the validation taken care of? That is the basic thing. The second thing is who is the manufacturer? So that has to be clearly stated in the manufacturing information card. And then the stability. Of course, any chemical is going to be uh, effective, unsafe, only if it is stable. Because when it is unstable, it may produce some unwanted reactive substances which are not good for the patients at all. So stability of the drug is very important. And the fourth one being the controls used for manufacturing the drug substance and drug product. The controls in the sense, any manufacturing company for that matter is expected to follow the good manufacturing practices. Like how good laboratory practices are implemented in the preclinical testing of chemicals, drugs, pharmaceuticals, etc. The good manufacturing practices, they dictate the purity of the product. The quality of the product is governed by the controls that are used in the manufacturing of the drug substance and the drug product. So these are the basic data that are to be included in the manufacturing information in the IND application. The third most important component of the IND is the clinical protocols and investigator information. Detailed protocols for the proposed clinical studies are required to assess whether the initial phase trials will expose the subjects, that is the test subjects, to any unnecessary risks. So, information on the qualification of the clinical investigators, who are professionals and usually doctors, who will oversee the administration of the experimental compound, is mandatory because they will be the primary responsible person who will oversee when the drug is dispensed and administered to the test patients. It is important to obtain informed consent from the research subject so that the patients or the volunteers are aware of the fact of the risks that are involved during the conduct of the clinical study. That they must also know that if you take a trial drug, it may not always end up in a good note, and there are chances of adverse events, sometimes serious adverse events, sometimes even death. So the research subject are supposed to be sensitized through an informed consent. Usually, it is issued also in the vernacular language so that the research subjects understand the risks involved during a clinical trial. And then this entire protocol and the investigative information, the informed consent, all these documents are reviewed by the institutional review board. Now, this board has the major responsibility of looking into all this information very carefully and very stringently so that nothing goes wrong and nothing goes wrong knowingly. So all the risk, the clarity of the protocol, the qualification of the investigator has to be very thoroughly ascertained so that the lives of the research subjects are not at risk because they're already risking their lives by registering themselves in a clinical trial, which comes with a lot of uncertainties. So there no known risk should be missed out by the approving authorities. So the IRB gives a very thorough and comprehensive review of all these documents before these protocols are approved for the clinical trials. The pre-IND consultation program of the FDA is very supportive to new researchers and new manufacturers who want to file their IND. Now, this program was initially designed to facilitate early communication between the FDA and potential sponsors who have new therapeutics or new molecules in their hand. Now, this advice can be requested if you have issues regarding what kind of data has to be generated to support the rationale. Do you want to test in humans? Because you just cannot go and do some testing in humans without a very strong rationale. Now, even if you want any support on the design of non-clinical pharmacology toxicology studies, you can ask them, including the design and potential uses of any proposed models that you have in hand. And you can also ask them if what are the data requirements for an IND application, and any support on the drug development plans and regulatory requirements for demonstrating safety and efficacy. So they are giving you a constant support right from the the planning phase until the execution. So you can make a use of this pre-IND consultation program. You also have the guidance documents for the INDs, but these are not legally binding. Of course, they indicate the current thinking of the FDA on a particular topic. There are many laws, regulations, policies, and procedure which you are expected to adhere to. So please make yourself aware of these before you file the IND. The Code of Federal Regulations are another set of documents which are to be uh, read, understood, and implemented in the respective portions of your IND application. Now, the regulations applicable for the IND submission process according the CFR parts include the drug labeling, the IND application, the INDA and NDA applications for FDA approval to market a new drug, orphan drugs, protection of human subjects because you will be using human subjects for clinical trials. So all the rights and the safety and any risk involved. That is why the consent comes in. So everything you can find those details there. The IRB has a major role to play in there. And financial disclosure by clinical investigators, that's a must because in the event of any serious adverse event, the clinical investigators are supposed to do a fair compensation to the family of the dead human subject. And the IRB has a very crucial and sensitive role in the initiation and the conduct of the clinical trial and approval of the clinical trials. They have a very sensitive role to play, considering that this is going to have a major impact on the human population. Since the clinical trials are the lead for the safety, any wrong decision by the IRB, any slightest mistake by the IRB is going to cost a human life. So the IRB has a very great responsibility in reviewing the clinical protocols which are submitted by the investigators. GLP for non-clinical laboratory studies is one of the important aspects of animal studies. GLP is good laboratory practices. This good laboratory practices basically came into existence because of fraudulent data that was generated by some laboratory in the past. Now, GLP is a very organized system wherein everything is planned. Everything, all your studies are planned by preparing, like how you prepare a study protocol for your clinical trial. A study plan is prepared for each animal study. Now, before the study, once the study is planned, the study plan has to be approved by the study director. Like how we have the principal investigator in the clinical trials, we have the study director in the GLP scenario. And once the study plan is approved, of course, if it involves the use of animals, then the Institution Animal Ethics Committee is supposed to approve the protocol prior to any testing. And following which, every step, right from the receipt of the test substance to the utilization to dosing the animals and all clinical sense, everything is documented in appropriate formats. And they are consolidated into a study report, which is also approved by the study director. And the entire process, the entire study, right from the preparation of the study plan until reporting and archival, the entire process is overseen by a quality assurance unit. So that is the basic difference between a GLP compliance study and a non-GLP study. The entire GLP compliance study is monitored by a independent quality assurance unit, which validates and assures that The study was fairly conducted and documented promptly and legibly, and everything was taken care of, and all the steps in the study were according to the study protocol. Any amendment to the study protocol is allowed, and any deviation which happened during the study has to be fairly recorded by the study director, and the onus lies with the study director. Now, in some of the countries, the study director is even liable to be legally punished if something wrong goes wrong with the drug, because the study data which is obtained from these experiments are archived for a period of 9 to 15 years based on the country in which the laboratory is stationed. So, in some countries, the study directors are legally binding. Suppose after 5 years of marketing, something is wrong with the drug which the study director actually did not pick up in his preclinical studies, then there is a possibility that he can be even imprisoned. So, regulatory authorities have a very important responsibility of looking at each aspect of the drug discovery or a medical device or cosmetic, whatsoever it is. The regulatory authorities have a very sensitive responsibility to look into the data so carefully, because it is going to end up in the human community at the end of the day. So, uh, as researchers, as professionals, we have it is our moral responsibility to conduct a fair trial, to conduct fair experiments, and to document them honestly, so that uh, it benefits the human population. In today's session, we had the opportunity to go through the steps involved in the regulatory registration of a drug. Now, as an innovator, you may have a new molecule in hand, and you may have some really good properties uh, with the drug that can potentially be useful to some human being. So, in order to take it to the market, you will have to go through all these regulatory procedures so that it benefits the end users. I'm sure you have had an overview of the drug regulatory process, and if you are an innovator, I'm sure you had some idea as to how you will go through this process, at least in a nutshell. Thank you very much, and good luck to you all.